Well, thank you, Jeff, for that very kind introduction. It's wonderful to be at this PLMI Thought Leaders Conference again. It's been a great pleasure to meet many of you over the course of the last couple of years. And it's always a pleasure to be here, as we saw from the first two speakers who were just spectacular today. So it's my pleasure to tell you a little bit about our work at the Institute for Systems Biology, working at how to take genomics and fill in a lot of the other dynamic information out of the blood, microbiomes, proteomes, metabolomes, clinical chemistries, and so forth, and how do we put this all together to help drive a future that's focused on wellness. So I'm going to start just by way of disclosure to mention that I am a co-founder of a company called Airavail, which co-founder with Lee Hood and Clayton Lewis, which is a company that's focused on wellness, and it partially funded some of the work that I'm going to show you here today and may license discoveries from there. So it's going to be a big part of the scaling, so I'm going to bring it up a little bit today. All right, so this is perhaps a familiar picture to some of you. This is Larry Smarr. And so Larry is one of the most prominent individuals of the quantified self movement. So for about the last 15 years, Larry has been measuring all kinds of things about his body. Larry has a few advantages over the typical patient in that he runs the San Diego Supercomputing Center. So he has an enormous amount of resources at his disposal. But he had kind of an interesting experience. So a few years ago, he had been measuring a number of things out of his blood, and it had been quite stable over time. And then at a period of moment, he saw this enormous jump in a number of parameters way beyond what they had ever been before. And so he got worried about this, and so he looked, he did some digging, leveraged some of his resources, and he said, based on the things I'm seeing changed, he says, I think I'm getting IBD, inflammatory bowel disease. So he goes to his doctor, and he tells him this, and he says, okay, how do you feel? He says, well, I feel fine. He says, well, then we can't help you. And so he goes to a few different doctors, and he gets basically the same response. And that's not meant to be a critique. That's just the system. They're not set up to deal with this data that they have not seen before. And so as he does this, a few months later, he has an acute episode. He ends up having to go into the emergency room. Sure enough, he has IBD. And so that's, of course, just an anecdote, or it's an N of one story. Right? Could have been wrong, could have gone the other way. But the interesting thing about Larry's experience is that if you take all those measurements that he has done on himself, he goes through the acute episode, he goes to the healthcare system, they do what they do well, they get him back to health, but those systems never return to the same state that they were before. They're much noisier now. So this gives us then a way to think about a theory of disease and a theory of aging, in the sense that as you go through your life, your body has homeostasis. You have regulatory mechanisms to maintain health in the context of a wide set of perturbations. But once you go over that threshold into disease, what can happen is that you have now altered that system. You have problems in one system. That system interacts with other systems. And so over time, you start accumulating a number of these, and eventually this leads to failure and disease. So what we would really love to do in the context of the program I'm going to talk to you about today is take this experience of Larry's, which is an N of one experience, an anecdote, and see, can we generalize this to lots of people, and can we put real science behind this kind of a predictive and preventive approach to medicine. So we all know that there are a number of issues that we face. One is that we have an aging population. Now, of course, the fact that we have an aging population is not a problem. Right? We worked hard to achieve this, right? This is what antibiotics are about. This is what clean water is about. It's what the healthcare system is about, right? It's what we wanted. So we have that. But the problem associated with that is that we also have a skyrocketing amount of chronic disease. And in fact, 86% of healthcare costs, according to the Centers for Disease Control and Prevention, 86% of costs of the healthcare system are for chronic disease. So one problem that we would say exists is that the current medical field focuses almost entirely on care after illness. Okay? Larry Smarr example. Data is based on population averages rather than on individuals, so starting to change. And we don't yet know, in most cases, when we may become chronically ill. And so, of course, the question that so many of us in this room are asking is, what if this were different? And how can we make that happen? So what you would love to be able to know is not just some risk of disease into the far future. As we know, these risks of disease in the far future don't motivate behavior in very significant ways. So what you want to be able to do, though, is know what's happening in your body at this moment that is limiting your wellness or that is biasing you towards something in the future, so that you can have an immediate actionable effect in the present to remove, to the extent possible, some of the scenarios that are leading to disease downstream. So if we look at determinants of health in the United States, there are three major factors. So the first is that about 30% of health outcomes for an individual can be attributed back to their genetics. 60% is attributable to behavior and lifestyle. And of course, preaching to the choir here, this is the Personalized Lifestyle Medicine Institute's conference. So we know that this is a huge aspect of driving health. And only about 10% of health outcomes through the course of a person's life is related to healthcare per se. Now that's an important thing for us to think about, because 97% of our healthcare dollars are spent after illness, on treating disease. So what we're really excited about is launching an area, a field that we call scientific wellness. And so there's a wellness industry. So many of us here in this room are associated with that. We have massive spending in healthcare, $3.8 trillion. But what we see emerging now is this field that we call scientific wellness. And the distinguisher there being that we have so much more ability now, and you heard some of this in the earlier talks. We have so much more ability to drive measurements, to get continuous data, to build models that are predictive, to come up with preventive strategies, so that we can focus on defining what wellness is in a quantitative way, and so that we can impact it. So that's the area of quantitative wellness, scientific wellness. So of course, all these things that I just talked about, kind of the vision piece, are extremely challenging, as we all know, And so the central problem that we face is complexity in biology and medicine, And it's enormously complicated. So we will soon have billions of data points about every single one of us related to our health, beginning with the genome, but, and super importantly, not ending there, where we have dynamic measurements from the wearable devices that are proliferating everywhere through the aspects that we can also bring in through what we call systems medicine or systems biology, which is that we can understand a lot of this data, not just in terms of big global machine learning type algorithms, but also in the context of biological networks whose functions we've been studying for decades. And so this is really a key component. So what we see is the emergence of P4 medicine. Of course, Lee Hood has been pioneering this approach to medicine and evangelizing about it for a long time. And so P4 medicine, as we call it at ISB, stands for a medicine that is personalized to the individual, that is predictive about what's going to happen, preventive to the extent possible, and participatory. And participatory is really a key aspect, and participatory is such a key aspect in terms of the audience that we have here, our community, because that's where the individual becomes the center of their own healthcare experience. And we would like to see them become the center of that, empowered by data, empowered by technology, and you're already seeing that happening. As Denny talked about in his talk to lead us off, you already can see the effect that having all that type of continuous measurement at home, that whole ecosystem that's growing up, is going to drive enormous change in medicine. So we see this P4 medicine emerging from three big trends. So one is systems biology, systems medicine, all the omics technology, genomics, and so forth that you can integrate together. Consumer-driven social networks, which we think are going to be huge pushers of change in the healthcare system. Healthcare system is big, has a lot of inertia, it has a lot of checks and balances in it for very good reasons, but patient advocacy is what's really going to drive the adoption of change, and then coupled with the big data revolution and the digital devices that are coming online. Okay. So in terms of conceptual themes for P4 medicine, then we tend to think of two major thrusts. One is the quantification of wellness, and understanding better what wellness is all about. I love Denny's characterization earlier. I hope I don't get this wrong. That medicine is the one field that doesn't fully understand its gold standard, right? Which is wellness. Very interesting statement from a leading physician. And then the other side, in terms of the conceptual themes, are disease demystified. So can we understand the mechanisms deeply? And so what we see coming out of this is we have a huge disease industry, right? The current healthcare industry, which is mostly a disease industry. We believe very strongly in the emergence of a wellness industry that ultimately will be even larger, we believe, than the healthcare industry or the disease industry. Okay, so this has all been sort of to paint a vision or a piece. And so a few years ago, Lee and I were working together with colleagues like Mauricio Flores, who is here, in something called the P4 Medicine Institute. And we were basically sitting around saying, okay, here's all these concepts. We're trying to get partnerships with different hospital systems and so forth. But how could we really drive change in the way that we want to? And we spent a lot of time working on this. And what emerged out of that in the end was a desire to launch a big longitudinal study. And so we were together. I remember Lee asking me, as we were talking about needing to get longitudinal data, how many people would it take to get statistical significance? What do we need, right? And so as we looked at that, we thought that if we could get to about 100,000 individuals, we would start to be able to see the emergence of different disease patterns, and maybe we could start to understand something about what's related to disease. We actually think we can get a lot of this in much fewer numbers, as I'll talk about in a moment. So we launched with this vision. So we published this piece in Science Translational Medicine in 2014, essentially announcing to the world our intention to recruit 100,000 people, get dense dynamic data clouds about them so that we could drive longitudinal information. I'll talk more about that in a minute. Okay, so the immediate question when you launch something like that, and I remember, as we were having that discussion, Lee says, all right, we'll launch the program. We'll call it the 100,000 Person Wellness Project, the 100K project. I said, okay, great. I said, okay, at current prices, you realize that's like $1 billion a year, right? $1 billion that we don't have, right? Lee, of course, went to lobby Congress to try to get $10 billion from them, but that didn't happen. So that led us to think about other ways that we could make this vision a reality. And so how to scale this. And that led us to start this company called Airavail. And I'll talk just a little bit about that. And so what was very apparent is that we couldn't launch this kind of vision within just the academic realm. So we needed to have an ability to scale this up in a way that would be economical so we could get it. Now, actually, our hope is well beyond 100,000 people, but to millions of people. So as I mentioned before, Lee Clayton and I founded this company this year, at least its current incarnation. And what was very interesting is we started to think about how we could impact this new approach to what we call scientific wellness, is that it became not just about the science, right? And Jeff read that quote from our article at the beginning of the introduction. But basically, it's not just about the science. We have to engage with a much broader range of individuals. So we were able to get connections with a group here that helps with outreach. We did an event for 700 individuals in Seattle the night before I flew here, just outreach into the community so that they get a knowledge about what's happening in science at the moment. Takes getting together all kinds of people in the computational realm, being able to put together software apps and such for individuals to interface with. Very crucially, and there's a ton of expertise on this in the room, in terms of behavioral coaching and how do we interface and take all this complexity of data and make it simple so that people can understand what to do, how to take actions, and so we can learn from that. Operations, of course, external. And then we were able to raise $39 million this year to get this started. Okay. So that's kind of the plan for how we're scaling up in terms of getting to that big number. So what we did over the course of the last year was we wanted to show basically how, if there was enough value, if there was enough information so that this kind of data would be relevant to people immediately. And that's really a central component of what we had to think about here. Because it's not enough to just think about the diseases downstream. That's why to really understand disease, you have to study wellness. And you have to study wellness because that's what gives you the power to give someone a unique and empowering experience today that then is going to enable us to have the rich and dynamic data that we need to come up with the discoveries related to the early transition states to disease. So to make that concrete, we took 100 individuals. We call them the Pioneer 100. Jeff Bland was amongst that Pioneer 100, as you heard. So 100 individuals to come through this experience with us over the course of the last year. Okay, and so why did we do this? So the one thing that I'll argue is that, at least in science, and I would say often in medicine, we often don't study what we most want to know. So I'll use myself as an example. So I have a grant on Alzheimer's disease from the National Institutes of Health. We study differences in the brains of individuals who have Alzheimer's and who don't. But when do we do that? After they're dead, right? It's not really what we want to know, right? There's some useful information there. But what we really want to know was 20 years before, what was happening in the body of the individual that got Alzheimer's and the one who didn't, right? What was different about them? And what we really, really want to know is, if we had seen the early signs in the person who was getting Alzheimer's, could we have stopped it? Right? That's what we really want to know, and that's the need for this kind of longitudinal study. So as we were thinking about how to launch this, the idea is that every person has at any point a health status. This could either be getting better or worse. It could be transitioning more towards wellness, more towards disease. And so, as we know, delayed diagnosis, working after problems have arisen, very expensive. Can you catch it in an early stage, and can you do something inexpensive to have an effect? Now, these economics very often don't work out very well for a single disease. Okay? And this was another reason why, when we launched this 100K project, we didn't focus it on one disease. But the economics change pretty significantly when you have a broad enough range that you're monitoring for all of those kind of things at the same time, okay? Because you're aggregating a lot of those delayed costs. Okay, so as we took this Pioneer 100, and it was actually 107 individuals. This was a 9-month study. We launched it in March of 2014, so around March through December. Was IRB approved, gave us the ability to do evaluation, look at the next phase. So we did whole genome sequencing for everybody. We did detailed blood, urine, saliva measurements. I'll talk about exactly what we measured in just a moment. But we did that 3 times, so every 3 months. Gut microbiome, again, 3 times a year. Continual self-tracking and lifestyle monitoring, mostly just using things like Fitbits. We do a lot of data integration correlation on the back end. And we do monthly coaching sessions for each individual in the program. Okay. So what this allows us to do then is to generate millions of data points for every single individual. So we have the whole genome. We have the lab tests, and this involves 1,000 different metabolites, a few hundred clinical chemistries. We do saliva to get cortisol, stress levels, gut microbiome, so we can see all the bacteria that are living inside the gut. And importantly, what we have here is a very unique, I think very powerful resource around the microbiome, because not only do we have the microbiomes, but we have a huge amount of other data about every individual to start to learn how to read and interpret the microbiome. So we're very excited about that. And then we have the quantified self information. So we take these quadrants together, and then we also do and add on top of that, and do so either through focus studies that we get funded through the Institute for Systems Biology, but basically go in deeper dives and add lots of different kinds of information on top of that where we can. For example, we did discovery mass spec proteomics out of the blood for the 100 pioneers. And so this allows us to get millions of data points for every individual and to create a data cloud around every individual. Now, of course, we don't just drop this data on top of people. So in essence, there's a big analysis team that goes through, tries to figure out what that data means, and it's all communicated to individuals via a wellness coach. And for the Pioneer 100, that was Sandy Kaplan, who was a terrific person, probably the most popular person on our study team, I would say, by a long way. People really loved working with here. And also we have a study physician who would oversee, and we also have members of our medical advisory board. Joe Pizzorno, for example, was very involved in our medical advisory board as well. So as we get this data from individuals, so there's this coaching, there's this feedback, there's this immediate benefit we hope to them, and then on the back end, for every person that checks the box saying that their data can be analyzed anonymously, then that data comes in a de-identified manner into ISB, and we can do deep dives around trying to understand new metrics for wellness, new learnings about early transition states to disease. So this creates a research portal. So the research portal has three big elements to it. One is breadth, because we do measure a lot of different kinds of data. And so we're able to integrate different kinds of data together in ways that haven't been done before. Frequency, right? So that's the depth. So it's not just one snapshot. You're getting this over time, over and over again. And its width, which we would say is the impact of recommendations. And this is really a key part. Because you have the coaching, because you have weekly email contact and monthly phone conversations with these individuals, you know a lot about what's happening. And many of them will carry out their own N-of-1 experiments. So they'll try different diets. They'll try different aspects of improving their wellness. They'll go through a transition state. And all of that information then is captured, so we have perturbations to the system where we can see what's changing and start to build that. And as you get those individual stories, you can start to aggregate them across this population so that you can start to get statistical significance and learn what's really happening. And so we think that's going to be really important for us. And so this then leads us towards being able to build out scientifically validated metrics. Okay. So I'm going to show you a little bit of the data from this pilot project, which is all getting written up into a paper that will be submitted here in the next 2-3 weeks. So one of the things that it lets us do right off the bat is to just do global correlation analysis. Okay, so nothing fancy here, but I do want you to consider this image just for a moment, because this is, the first reporting of measurements of the genome, microbiome, proteins, metabolites, clinical labs from the same people at the same time, over the course of a year. Right. So what that means is that even using pretty basic statistical approaches here, we can already start to see connections that have never been measured, never been looked at before. Okay? And so this, I think, is really fascinating. So every one of those lines represents a correlation between something in your microbiome to something in your blood, something in your genome to something in your proteome, and so forth. And I'll talk about a few of those as we go forward. And so this, I think, is just the beginning of shedding a light on a lot of the dark matter of biology. So we start to have the data that we need to start understanding the complex system that is our body, right? And we need massively more than this, right? It's an enormously complex system. Okay. All right, so we can think of 4 different transitions that we might be interested in. So from more to less wellness, right? From less wellness to more wellness, from wellness to disease, or from disease back into wellness. And so we'll talk about a few of these kind of transitions, again, coming back to this data that I was mentioning before. So one of the things that happened in the study right off the bat, and maybe for a number of people in this room this won't be a surprise at all, but one of the things we wondered is how many, would everyone have something actionable? Like when we give this data back to people, is there something to do for everybody, or is that for a minority of people? And in fact, in our study, what we found was that there was for 100% of individuals, they had something actionable, something that they could do that they could make a difference on in their health. So we started by just looking at the clinical labs. And as I start walking you through the results, I'm going to start from the simple and go to the more complicated. Okay, so we'll start simple. We'll get more integrative as it goes along. But just from the clinical labs, 91% of individuals had some sort of nutrient abnormality. 59% had some sort of markers that were out of range, say for cardiovascular disease, 54% for diabetes, 68% for inflammation. And this was in a presumably well population. Okay, so there were a lot of elements that they could look at or evaluate. Some of these areas, of course, we know better than others. But here was improvements that we saw in cardiovascular improved by 6%, diabetes improved by 33% in terms of the individuals that were classified as being pre-diabetic. I'll talk about that in detail in just a minute. Inflammation improved by 12%, according to the markers, and nutrition by 21%. So we saw over the course of 9 months a significant impact on a number of areas for these individuals. In terms of diabetes, now diabetes, of course, and it's been alluded to before because it is the obvious, poster disease for what we want to be able to have, because we do have an understanding in the community of what a pre-diabetes state is, and we know how to back out of it, right? And there are lots of programs that can help do that. We saw this work very well in ours as well. So by using HbA1c, fasting glucose, HOMA, insulin, we saw improvements in insulin resistance of better than 50% for the individuals in terms of who was out of range over the course of our study, and by 38 and 19% for HbA1c and glucose, respectively. So we're able to see some nice elements. Another one, and this is very interesting because, and I was just chatting with Joe Pizzorno before coming up, about the importance of toxicity, right? And toxicity is a major player in a lot of diseases. We do monitor for certain kinds of toxicity in our study. One of them is heavy metal toxicity, and here is the case of mercury. And we actually had 2 individuals, we had 3 individuals that were quite out of range in terms of mercury. 2 of them had the same source, which was eating a lot of tuna sushi. And so eating a lot of tuna sushi, this drove having a lot of mercury. By just switching that from tuna sushi to salmon sushi, was able to drive, as you can see over the course of just 3 months, the mercury level in the blood to half. So that's a very simple sort of dietary recommendation. Was doing, setting the stage for damage, able to just back out of that very easily. And one of those was Jeff Bland, and the other was Ed Lazowska, who's the yes. So there you go. So early transitions identified now. So that was all very simple stuff, right? Simple labs, just monitoring, simple steps to take. So let's just start adding it to complexity. Let's talk about just taking 2 of the data types together. So here, of course, what we're interested in is environment and genetics, their interplay, and how we can drive learning and actionable recommendations, behavior change for individuals. So we'll talk quickly about one pretty dramatic case in this, and there are going to be, again, in this audience, people that will be familiar with this. So one of our individuals was having cartilage breakdown in his body, had arthritis in his knees, and he didn't know why. He was on arthritis medication, didn't seem to help. He had concierge medicine on both coasts, but didn't really know what was wrong. And I think the reason for that was just that, in medicine, it's usually sort of a hunt and peck type approach because you've got a limited amount of data. You try to figure out what's the next thing. You get a limited amount of data you see. So we took this 360-degree view of him, measured everything that we do. And one of the things that jumped out very dramatically was that his ferritin levels in his blood were just extremely high. And of course, if you know anything about that, that is a warning sign for something called hemochromatosis. We went into his genes, which is one of the most common, actually the most common genetic disease for Caucasians. So we went into his genome. Sure enough, he was homozygous for that disease. So you could see it in the genome. You could see that it had manifested. We expressly are not in the business of practicing medicine. So, of course, we just refer him back to the healthcare system. They make that diagnosis hemochromatosis. He gets treated for that. And sure enough, when he did that, his ferritin levels dropped way back down, as you can see here. And the cartilage breakdown in his body stopped. And the arthritis in his knees went away. And so hemochromatosis has a very negative set of symptoms that you can drive, ultimately liver failure and culminating in death. And so I just want you to contemplate, it's a simple example, but just contemplate for a moment that with very simple, with just knowledge, it lets him take a very simple step to avoid years and years of suffering and ultimately a terrible disease. And the simple step that he had to take, and probably people in here, you know how hemochromatosis is treated? You donate blood. You donate blood. So if you donate blood and you get rid of that excess iron, you get rid of all those downstream negative consequences. It's easy to do if you have the knowledge that tells you that you need to do it. I think it's a beautiful example of what we want in terms of preventive medicine. The other thing, though, that I have to laugh about a little bit is that we've done this very sophisticated, huge data, lots of computation analysis, and we found the one disease that you can cure by leeches. That was my first thought. I'm like, wow, that's kind of amazing. It doesn't stop there. Come full circle. The other interesting thing is, so you can see here, if you look on the, that we have our individuals. If you have 0 copies of the gene, you tend to have low ferritin levels. You can see that in our population. If you have 1 copy of the gene for hemochromatosis, they're in the middle. And if you're at high, then you've got that really elevated level. There's only 2 individuals there, right? And I should just keep reminding you, this is the pilot study of 100 people, so that we'll have a lot of kind of smaller sample sizes. It's the whole point of scaling big. Now, the interesting thing is if you look at those 2 individuals that are homozygous, they're actually radically different. So you've got the person that had the disease who's super high. You've got the other person who is not so high. This is a woman. She's premenopausal. Okay, so as long as she's premenopause, it's very unlikely that this disease will manifest. But, and again, this is getting to the preventive and participatory aspects of this kind of medicine. She now knows to be on alert that post-menopause, it's very likely that this may elevate. But again, very simple. Monitor your ferritin, and if your ferritin gets high, then donate blood. And just work with your healthcare provider and work with her physician and just keep track of it. And so she knows to do that. So now it's very unlikely that it will ever become a problem in her life. Right? So knowledge is empowering. Okay. All right, so let's step up a little bit more in complexity. So that was just a single genetic variant, right, associated with hemochromatosis. But now we can take all these different variants and start putting them together. So we can predict individual risk for diseases based on summing up across various different variants for between 50 and 70 different diseases now. And I should say you can do that for disease. You can also do it for quantitative traits. And I'm going to show you one of those right now. So for one of these traits is that you can predict cholesterol levels in the blood from the genome. Okay? And I'll show that in just a minute. So basically you can sum up anyone's particular risk. And then you can bin them in the population, whether they're at low risk or high risk for having high LDL cholesterol or low HDL cholesterol or whatever. And so I'm going to show just one simplified picture here. This obviously isn't from all the 100. This is a subset that's for a family within the Pioneer 100. And I just want to show it because, and I want to be clear about this. Like, this is a small data sampling, so what I'm going to show you here, I wouldn't, this needs to be better powered, but I just want to give you a sense for what we're thinking of and where we're going and what we're excited about. So if you build this, and the correlations hold up beautifully across all the data, but you can't see it quite as clearly as what I'll show here. So basically, you can get a very good correlation for LDL cholesterol. In the family, you get 0.85 correlation, which tells you that genes dominate the LDL cholesterol levels in the blood of these individuals. Okay? HDL, which is as is very well known, is less predictable by genetics, much more influenced by behavior. And so that's where I want to focus just on a couple of things. Okay, so people know that, but now with these genetic risk profiles, you can start to ask different kinds of questions. Okay? So one of the first questions that you can ask is you can look at people who are deviated away from this line. Okay? So you see that one person who's really high, right? Who's really high there. So their HDL, their good cholesterol is, looks great. And what's interesting out of this cohort is that that's our champion triathlete. So very, very good shape, super athletic, takes amazing care of his body. And so you see that movement away from genetics, right? We have another individual who's very low there, and so the question is, all right, why are they low? But you start to have an understanding of not just what your blood measurement is, but what is your blood measurement in the context of what you might expect from your genetics, okay? Early days, we have to, this has all got to get proven, but we start to be able to ask different kinds of questions. The ones I'm really interested in are these down here. And the reason I'm so interested in them is because here, all right, they all have the same low level of HDL, okay? All right, and so we would treat them as being the same in the current way that we would do the healthcare. But what's fascinating is that for 2 of these individuals, we predict that they're kind of right around what their genome would predict. And for this other, they're very far from where their genome would predict. Okay? So one of the things that we're exploring now is how relevant is that information to understanding your health? And there's all kinds of different ways this could be interpreted, right? It might be that if you're close to your genetics, maybe you're actually not that bad off. Maybe that's just homeostasis for you, right? That's one interpretation. A second interpretation could be the opposite, right? Which is that, no, those are just bad genetics in today's environment, and that you're going to need probably drugs and help to get up much more so than the person whose genetics supposedly would enable them to move up much, much easier. Okay? And so I just want to give you a taste of what we're thinking of here, because as you start to integrate this data, you can now segment and ask and personalize these kind of measurements in much more powerful ways. Now, you would never conclude whichever of those hypotheses is true from this scant amount of data, but it's so evocative of what are the kinds of questions we can ask, and as this scales to 10,000 individuals and beyond, to 100 to millions, we're going to be able to parse and understand that kind of information about how genetic predictors are related to the interpretation of your health data in much, much more important and more powerful ways. And so that is it's just a taste, but I think that this is really a huge growth area, certainly for the future, which is to fill in all that information between, I have this gene, it's related to this disease. All right, well, what's happening in between? Okay. All right, so now let's talk about transitions identified, again, from integration of multiple types. So here let's just look at genetic and microbiome. Again, it's a small sample size, so I'm not trying to give you too much. I wouldn't read too much into the specifics, but I want to make the general point. Okay, so the microbiome, right, is highly diverse across individuals. This is the rates for the high-level phyla from ours, and Bacteroidetes and Firmicutes, like in every study, come up as being the highest. And we see pretty strong variety at that highest level. You have people that are essentially have almost all Firmicutes, and you have people that are vast majority Bacteroidetes, and it goes back and forth. In classic overfitting, right? So you wouldn't want to read too much in this, but these are 2 of the best computer scientists in the world. So, so I don't know. It's like Firmicutes are good for that. Right? That would be the classic overfitting specific detail to not read in, but general trends we want to talk about. So one of the things that we are very interested in, though, is coming back to this notion of genetic risk. So we've built just a genetic risk profile, just pulling data from the literature, that bins individuals into either very low, low, medium, high, very high genetic risk for Crohn's disease. Okay? And so as we look at that, we can then say, are there any differences in the microbiomes in these different categories? Okay? Again, small sample size. But basically what's interesting is that the verrucomicrobia, as you can see here, go from 0% to 1 to 2 to 4 to 7% as a function of just genetic risk for Crohn's disease. Okay? So it's a small sample size. I was giving this to a friend of mine who's actually the director of bioinformatics at Mount Sinai. And we were in a meeting, and I showed him this, and I'm like, it's preliminary. He's like, oh, yeah? He says, well, I have Crohn's disease, and I just got my microbiome done. And I was like, oh, okay. I was trying to pitch him on this stuff. And so he pulls it out, he opens his laptop, he looks up his microbiome, he looks up verrucomicrobia, and sure enough, he has 8 times the amount of verrucomicrobia that someone in the that a normal person has. And so that's, again, just another one, but it got them excited enough. I'm actually going to fly to Mount Sinai tomorrow, but we're looking at, can we launch actually a program together so that we can get huge numbers of individuals that have Crohn's disease and start diving into some of these relationships in a lot of detail as you would need to. So here, just sort of an evocative story. But maybe there's something there, and we're going to dive in and find out. Okay. All right, so now just getting into the last phases here, I'm going to talk about a little bit more sophisticated models, just the tip of the iceberg. We've got like hundreds of these things, but I'll just show you a few. So one is that, as I mentioned, we can do very basic correlation analyses. Okay? But one of the things that's really interesting here, so you can take the top 2 are just sort of controls. So these are from published studies, and it says here, this is a predictor from your genome for this particular trait, and this is the actual measurement in the blood. And you can see that you get correlations around 0.5, p-values around, I guess, adjusted p-values 10 to the minus 13 or so. And so you get these very statistically significant relationships. But what you can see here, down here, then, are some of the new ones. So one interesting one is if you look at genetic risks for having an elevated blood pressure, for example. If you have low genetic risk, at least in our population, every individual that has low genetic risk for blood pressure, not a single one of them is on hypertension medication, right? And that's what you see here. Right? This is another thing that we're rolling out now to different hospital systems. In the upper, you have a distribution. Some are on and some are not. But if you're low on this score, essentially nobody is on it. And so you can start to see, okay, genetics are having a pretty important role there, obviously. Another one, and this is one that we find very fascinating, is that if we look at this molecule related to inflammatory bowel disease, so we get the same sort of correlations as what we've seen up there, so a very strong signal, okay? And so as we're looking at this, basically what this means, and the molecule in this case is a cystine. And so what's really fascinating is that if you do case control studies for inflammatory bowel disease, cystine comes up as one of your potential biomarkers. It's very differential between case control. Now what's fascinating is that we don't have any case control. We don't have any disease. All we have is a predictor for your genetic risk of disease. And it comes up very strongly. Okay? So that means we found it without actually having anybody in the study who had the disease. Okay? So that's really fascinating. And there's a whole, again, because it's early days, there's a lot of different interpretations. One is that case control study is in fact not measuring anything that is related to the disease having initiated. Because remember, none of our people have the disease, and they range in age from 20s to 88 plus. Okay? And I say it in that weird way for PHI reasons. But basically, so you have a huge age range. So what this would tell you is that cystine very unlikely to be a candidate as an early marker because it is a pre-existing difference. Now it is also related to oxidative damage pathways, and so there is a potential mechanistic linkage between these. So it could be that it's related to the genesis of the disease. You need more study. But again, I think what's so interesting is that as you start just filling in some of this detail, I think that this kind of a program where you have this longitudinal information coupled with genetics is going to be absolutely key to essentially all biomarker studies in the future. Because I think that as you go in, to not understand the genetic differences between your population and try to interpret results, I think that's really a mistake. And so what this kind of information is going to let us do is it's going to mean, because consider how every case control study is done, right? You have your cases, you have your controls, you match them by age and sex and every other variable that you think is relevant, and then you get a set of people that have the disease and a set of people that don't. Well, it stands to reason that the set of people that got the disease were more genetically predisposed to that disease than the set that didn't. And wouldn't you like to know, because of their genetics, what are all the things that you would expect to be different just because of their genetics? That's going to tell you about things that are pre-existent and not related to the onset of that disease that you're studying. Okay, so I think that's really a key point. We're going to drive that very strong in the next year. Okay, just about done. So here we have a model. This is a linear regression model, so more complicated, so I won't walk you through all of this. This is just coming up with predictors from the data for, in this case, EPA, so one of the omega-3 fatty acids. And one of the things that's sort of interesting here, so these are just the coefficients on the model. So whether or not someone takes fish oil or not, for example, is essentially not predictive of the amount of EPA in the blood. What is predictive is the amount of mercury, right? And remember, mercury is a proxy for eating fish, right? So that does come up. And then what we get as a very strong predictor is vitamin D. And so, there's at least a putative link here between vitamin D and EPA. And we're just starting to dive into this. And again, I really want general level trends rather than the specifics here. We do dosing of vitamin D because it was a Seattle population. It was very, we were very vitamin D deficient. Although Jennifer, who is our health coach, likes to point out that that's not a Seattle phenomenon. It's a little bit of a misnomer. It's actually very widespread. But so we get a dosing effect. And the other interesting thing here is that, so we do two kinds of correlations. We do correlations across the population. We also do what I call delta correlations or time correlations, which is just looking at correlations between the change in an individual between different rounds, round 1, round 2, or round 2 to round 3. And the interesting thing here is that we also get a strong correlation here between the change in vitamin D and the change in EPA. So again, it's not just that they're correlated across individuals, but when one goes down in an individual, the other tended to go down as well. And so we have, and again, this is just the tip of the iceberg, we have 10,000 such correlations, but basically, there's a lot that we can just start to understand and puzzle out, and it leads to lots of hypotheses, which is why we think that and this is why we're so interested in partnering with different organizations, certainly through ISB, so that we can launch in and dive into some of these that are really the most promising. All right, so just coming back, that was just to show you just a very few of these massive number of correlations that have been pulled out. And then since the title of our program here is on cancer, I'm just going to make 2 points about ways that we're interested in getting and interfacing on cancer. So one is that we think and would love to launch, we're looking for partners around this, which is a cancer wellness program that takes the kind of things that we've done in the Pioneer 100, tailors them to the difficulties that people go through during treatment and after treatment. Because even if someone goes through chemotherapy successfully, right, you may get rid of that tumor, but you have damaged an awful lot about your body. So why don't we look at that in great detail and try to figure out how do we mitigate that from happening in the first place, and then how do we get you back to your blueprint of your wellness. And of course, the other aspect is then to look at cancer incidence and what can we do in advance of cancers to try to keep them down in numbers. So with that, I'll close. We see that we just want to enable individuals to be at the center of their health care. As we talked about, and I hope you saw a number of examples of this, knowledge really is power. Having knowledge enables you to make decisions to maximize your wellness today and aids in avoiding disease tomorrow. And with that, let me thank some of the people who have been involved in this study. Lee Hood, of course, who has been just a fantastic partner and mentor through all of this for the last decade. Sean Bell, who was our business director, who did absolutely a ton of work. Jennifer Lovejoy, Sandy Kaplan, Craig Keebler, who really helped us on and led out the engagement with the participants, the individuals, all of those aspects. A great data analytics team, some of the best people in my lab, Andrew Magis, John Earls, who joined on, Gustavo Glusman, our medical advisory board, and all the other people. Thanks.